Let's now invite Tiffany Vora, who's a doctor in physics, a non-resident member of Geotech of Atlantic Council, and a member and vice president on Biology University in Singularity University. She has a licentiate and a BA in chemistry in the University of New York and a doctor's degree in molecular biology in Princeton University. Dr. Voda worked in the pharmaceutical industry. She has been on the American University of El Cairo, and she's been a teacher of bioengineering in Stanford University. She founded 2 companies focused on communication strategies for scientists and experts. She works as a consultant in several companies. She's a member of the Women's Homeware Bank Project, and she's part of the leading team of the Space Humanity Lab. Let's invite once again Débora Slotnisky to start her presentation, How Women Will Program the Future of Life.
Thank you so much for that wonderful introduction, and thank you so much for inviting me to join you today from my home in California. Thank you as well for enabling me to speak English during our conversation today. My Spanish is pretty good, but it's not science good, so I'm just delighted to be here with you all today. And as we've just heard in that great introduction, today we're going to be talking about biotechnology and how women are taking a leading role in programming the future of life. Now, it has felt the last year and a half like a deluge of bad news. I'm sure you've been feeling this as well. But there's going to be good news coming because the world is changing. I want to make sure that you are looking at what I'm looking at. Perfect. Okay, the world is changing because the boundaries between living things and technology are starting to break down. We are entering a new era in which the living parts of you are accessible to ever cheaper, ever faster, ever more powerful technology, computers, sensors, and even more than that. Today, we're in an era where the biology is increasingly being the technology. And I want you to remember that message as I'm telling you about this framework that's called the Digital Biology Framework. And so to start out there, just in case you don't spend every day thinking about biology like I do, just as a quick review, all life on Earth is related, and all life on Earth, including you, is made of cells. Living thing stores what's you can think of as its operating manual. We call that the genome. It's a collection of DNA that's basically the operating instructions for building and then running that living thing. Those instructions are organized into groups called chromosomes. Now in humans, we have 23 pairs of these chromosomes. That's where the famous company name 23andMe comes from. So now let's think about just one of those operating manuals, just one of those chromosomes. And if we zoom in even further to that chromosome, we get down to the level of the famous DNA double helix. It's a chemical molecule that's made up of 4 chemical building blocks that, for short, we call A, C, T, and G. And those A, C's, T's, and G's are the coding system of life. It's the coding language of life. Computers store information as zeros and ones. Living things on Earth store information as A, C, T, and G. Now your genome, the human genome, has about 3 billion of these A, C's, T's, and G's. And it's the order that stores the information because there's only 4 letters in that alphabet. And it turns out that 99.9% of your DNA is exactly identical to the DNA of every other human being on Earth. It's the 0.1% of those A's, C's, T's, and G's that make you, you, and make me, me. So as I think you can see, knowing that order of those A's, C's, T's, and G's is really, really important for all kinds of industries today, for health and medicine, for agriculture, for energy, and as you're going to see in a little bit, also for some really exciting applications that are starting to come down the line. Now, you may remember from school that the two men who shared the Nobel Prize for this discovery of the structure of DNA were named James Watson and Francis Crick. But what you might not know is that Francis Crick and Jim Watson and Maurice Wilkins shared that Nobel Prize for solving the structure of DNA in 1963. But they never would have been able to do it without a photograph that was taken by this woman. This is Dr. Rosalind Franklin. She was an absolutely brilliant X-ray crystallographer. X-ray crystallographers take molecules, they bombard them with X-rays, and then they use really fancy mathematics to figure out what is the shape of that molecule. Well, one day, Dr. Franklin was giving a talk about her work. She showed this picture, and sure enough, Watson was in the audience. And the second he saw this photograph, he knew that DNA had to be a helix. That was the key breakdown. Now, 10 years later, when Watson and Crick won the Nobel Prize, they shared it with Maurice Wilkins, Rosalind Franklin's boss, not Rosalind Franklin, because she had already died. And it turns out you can't win a Nobel Prize if you're dead. So this is a woman who has been called the Dark Lady of DNA. I'm showing you my copy of this book that is a biography of Dr. Rosalind Franklin. And it's really important, I think, to know her story, because what we see over and over is that women make really important contributions to the sciences and to the life sciences, but sometimes they're overlooked by the systems that we have in place. So going back to this idea of digital biology and the source code of our DNA, that means when I think about DNA, I'm not actually thinking about a helix. I'm thinking about source code. I'm thinking like this. And this is really powerful because anything that we can do with DNA code is the same type of thing that we can do with computer code. And I want you to remember that because we're going to come back to this idea in a little bit, DNA as code. But you might be asking yourself, okay, so there's all this digitized DNA information out there. Where does it come from? The answer is it comes from a set of technologies that are called DNA sequencing technologies. And it turns out that DNA sequencing is a beautiful example of an exponential technology. So here, what I'm showing you is the price performance curve of DNA sequencing. Now, this is a semi-log plot, so the y-axis is a log scale. That's why Moore's law here is a straight gray line. So you might remember back in 2001, that was when the draft of the human genome was published simultaneously by a government group called the Human Genome Project, and also by a private company called Celera Genomics. And back in 2001, just to get that draft of the human genome, it cost about 2.1 billion US dollars. You can see then that the price performance of DNA sequencing then approximately followed Moore's law until 2008, when two things happened. A different type of chemistry was invented for the DNA sequencing, and the computation changed. The computers got much more powerful and started analyzing the data in a different way. And once that happened, you can see after 2008 that the price performance of DNA sequencing radically outperformed Moore's law, all the way down to 2019, which is the last year we have really good data for, when the cost of sequencing a human genome had dropped to 900 US dollars. Now, as a reminder, in my country, in the United States, 900 US dollars is cheaper than an X-ray costs. So that says a lot about a lot of things, and it tells us just how transformative this technology is poised to be. Because in the space of 20 years, we went from DNA sequencing of a human genome costing as much as a fighter jet to costing the same amount as 2 VIP tickets to a Taylor Swift concert. Huge, huge change. And in fact, we think that in the next few years, that price is going to go down even further, where the consumers are going to be paying about 100 US dollars, but the marginal price, the price to the company that's doing that, is going to be pennies on the dollar, meaning that it could be cheaper to get my DNA sequenced than to flush my toilet here in California. That's pretty remarkable. On top of that, the machines have changed a lot. So I'm showing you here a picture of a DNA sequencer from 2001. It was about the size of a refrigerator and cost about $1 million US. Now, today, 20 years later, we still have refrigerator-sized DNA sequencers that cost $1 million. They are way more powerful than they used to be. But we also have DNA sequencers that look like this. This is a handheld DNA sequencer from a company called Oxford Nanopore in the in the UK, Oxford Nanopore. And that handheld DNA sequencer plugs into a laptop. Now, there's another version of this called the Smidge Ion that actually interfaces with your smartphone. So what that means is, in theory, you could have a DNA sequencer anywhere where you have a pocket, a little bit of electricity, and an internet connection. Really radically transforming things. And we've seen how important this has been during our COVID pandemic. And what this suggests is that we are coming up on a radical shift in healthcare, also in agriculture and in lots of other verticals. But healthcare is interesting because right now, today, you basically wait until you get sick to go to the doctor. You wait until you have headaches or your vision is changing before you go to the doctor. Your doctor does an MRI and she finds a tumor for you. What I'm trying to tell you is that with these various biotechnologies, which unfortunately we don't have a lot of time to talk about today, but together what they're doing is moving us into an era where we can switch from sick care to true healthcare, data-driven, highly personalized, predictive and preventive care that comes based on actionable insights from our own bodies. Now, there's amazing examples of this all over the place. I want to tell you one about a company called Grail. So back in 2013, one of the big DNA sequencing companies, it's still out there, it's a company called Illumina, was running a set of experiments where they had their, a pathologist, their director of the lab, was looking at the blood of pregnant women, and she was using it to try to design a test for fetal abnormalities in the pregnant women so that you wouldn't have to do an amniocentesis to see if there was something wrong with the baby. And instead, what this pathologist saw was DNA fingerprints that made her suspect that some of these pregnant women actually had cancer. And sure enough, that was the case. And this woman went on to found a company called Grail, spun out of Illumina, and it was recently, just a couple of months ago, bought back by Illumina for $7 billion US. Now, here's why I'm telling you this story today. Just a couple of months ago, as Grail put out an article talking about this great story of their founder, and they didn't even use her name. So I'm going to say her name for all of you now. This was Dr. Meredith Hawks Miller, who was the pathologist who was looking at the DNA data and said, wait a minute, there's something strange, there's something else going on here. Nobody else wanted to listen to her, but she persisted. And sure enough, this turned out to be a billion-dollar idea that could potentially radically change how we do testing for cancer. And this was the correction that Grail printed later, because actually Dr. Hawks Miller's daughter said, wait a minute, you didn't even say my mom's name, and she's the one who did all this work. And so Grail did apologize for having left her name out of the original story. So it turns out that digitization has happened for more than just DNA. We can actually digitize a great deal of biological information. And I want to draw your attention to just one of those things, which is proteins. So I told you that DNA is the information storage molecule in our bodies, in all living things. Well, proteins are the molecules that actually do things. They carry out the instructions of the DNA. They're incredibly important. And the way that each of these proteins has a particular function is because of the protein's structure. So if we know the structure, then we know something about the function. And so for many, many, many years, one of the most important things you could do as a molecular biologist or a biochemist was to solve the structure of a protein. And that could take you all 6 years of your PhD, just solving for one molecule. Well, you might remember late last year, the company DeepMind published an algorithm called AlphaFold that was able to solve computationally an AI could solve the structure of a protein that previously it was taking humans years and years in order to do. And just a couple of months ago, another team at the University of Washington published another algorithm that's able to do this as well. And so now what we see is just in the last couple of months, these gigantic databases with more than 300,000 protein structures in them that were solved by an AI. And I think what's most exciting about this is not only are these data open source, but the algorithms are too. So I think of this type of technology not as going to be taking the jobs as scientists, but instead of giving us superpowers. I can't tell you the number of labs who woke up in the morning, saw this news, and instantly went to go download that software because they knew this was a superpower. And so that brings us to another point that I want to make today, which is as we're moving into this era where computers are giving us more and more of these superpowers to do great work, including great science, it is so, so important that we teach our girls to code. If you only take one thing away from this talk today, it's that. Teach our girls and our women to code, because this is the real frontier and where we're going to be seeing amazing progress in biotechnology, life sciences, agriculture. We're going to be using these superpowers from computers. So I just told you a bit about how biotechnology and molecules are coming together to have some really fantastic innovations. Let's zoom out now and look at cells, because we can also be using cells as the building blocks of technology. Let's look at just one example of that. So think about your food, right? Our food, our protein, our meat, our fish comes from living things that are killed and then we eat them. But instead, there is a whole new movement going on where what folks are doing now is taking some of those cells from a living animal, you don't have to kill the animal to do this, growing them in giant tanks like the ones that we would use to make beer, and then what you get is lab-grown meat. This is also known as cellular agriculture. It's an incredibly exciting innovation space that is coming very close to market, so you should keep your eyes open. And it's exciting because it means that we are able to get nutritional and frankly the enjoyment of eating the foods that we are used to eating and that taste really delicious to us, but without torturing and killing billions of animals and without having the massive environmental footprint that we know industrial agriculture has. This has the potential to be win-win. And I can even imagine a future in which we do this cellular agriculture on the counters in our own kitchens, not even just in giant warehouses all over. And when I think about exciting things like going to Mars or other planets, I think about this as well as being a really empowering technology. Now, I first truly understood the potential impact of cellular agriculture when I went to a talk that was given by this woman, Dr. Sylvia Earle. Dr. Sylvia Earle is a National Geographic explorer in residence, and she was the former head of the National Oceanic and Atmospheric Administration in the United States, the first woman to run that position. And Dr. Earle really made me understand that if we are serious about protecting our oceans and protecting our planet for our children, their children, and their children, then the way we make our food really has to change. And cellular agriculture is a great way to do that. And so as Dr. Earle pointed out, never before in all of human history has there been greater opportunity or need for young women in science and engineering and in all human endeavors. Women are going to be crucial to this moving forward. So now we can zoom out one more step and say, well, what happens when we take biotech and we try to make whole living organisms with them? Now, remember, we talked about AI not too long ago, and we're going to come back to AI here. 2020 was a disaster of the year, and I want to tell you about my favorite scientific study that came out in 2020, and it was this one. This was a study where a group of researchers decided that they were going to ask an AI, train an AI, in order to design living robots. And they said, okay, AI, you get two building blocks. One building block that holds still and one building block that can move. Now give us designs for building a robot that will move with only these two kinds of building blocks. And if you program your AI right, it does exactly what you ask it to do. And so this AI returned a bunch of these plans for making these moving robots out of only 2 building blocks. And then the researchers went, they chose some of these plans, and they went and they got 2 types of cells from frogs, as it turns out. One type of cell was a cardiomyocyte. So that's the cell that makes the heart beat. That was the moving cell. And then the other kind of cell was ectoderm, which is a cell that doesn't move the same way. And then they built little tiny cellular robots based on the AI plan. And when they did this, sure enough, these little robots, which they named xenobots, moved the way that the AI predicted that it was. And again, all we did was take 2 biological building blocks, put that together with the superpower of AI, and we have now living robots. It's really very exciting. I can't wait to see what comes from this research. I think it's a really exciting field, and I can't wait to see what xenobots do. So let's go back to our digital biology paradigm, because it turns us into engineers of life, not just observers. So for example, we can move great big blocks of code around. That's called genetic engineering, and we've known how to do that since the 1970s. We can take a piece of code out of one organism and put it into another organism. That's also genetic engineering. We can write DNA code from scratch. That's synthetic biology. And we can do cool things like debug the source code of life 1 letter at a time. And that is gene editing, which I'm sure you've heard of. And we're going to come back to that in a minute. But so let's go back to this idea of being engineers of life. Think of a computer. An app is a program that does one thing, right? It carries out a task. It takes some kind of input, does something to that input, and then makes some kind of output. Well, you can think of living things as doing the same thing, taking source code as input and then running it and creating an output. And I know that you have experienced this in your daily life from just watching the last 1.5 years, because this synthetic biology paradigm is the basis for RNA vaccines, like the Pfizer vaccine and the Moderna vaccine against SARS-CoV-2. So the way that worked was back in January 2020, when Chinese researchers uploaded the first sequence of the coding instructions for SARS-CoV-2. It has an RNA genome, same idea. Then what scientists at a whole bunch of companies, including BioNTech and including Moderna, were able to do was to take that code, digitize it, and then say, okay, which parts of this encode for parts of the virus that we think the human body is going to be able to recognize and mount an immune response against. And so if you've gotten your COVID shot, and if you haven't yet and it's available to you, I highly recommend you go get your vaccination as quickly as possible. I got Moderna. I was very excited about that. That vaccine, an RNA vaccine, is taking the instruction manual for building only part of the virus, which is why the vaccines can't make you sick. That code gets loaded into you. You're the hardware. Your body then boots up those instructions, makes a little piece of one of the proteins of the coronavirus, and then your immune system learns to recognize that, and that's how you're immunized. So this is happening right now. And in fact, the founders of BioNTech are a husband-wife team who are living in Germany. They're of Turkish descent. And Dr. Özlem Türeci has been recognized as potentially someone who's having the greatest impact on humanity in the last 1.5 years of anyone else on Earth. And I have to say, if you are going to impact 1 billion lives, here was a great way to have done it. So we thank her very much for what she's done. And in fact, these companies believe that these RNA vaccines are going to be important for so much more than just COVID. So if you think that there are cancer vaccines coming, I would keep an eye on this space. That's what I'm doing. Now, I said that DNA is both a parts list and a coding language, which means that you can add parts to the list and you can take parts out. And if you add a part to that parts list to the genome, the cell will build it for you, just like our bodies are building the protein that we get the instructions for from our COVID vaccines. And so this is really exciting because it means that the genome is kind of like the chassis on a car. You can plug and play different elements to get different results. So maybe I want to put in a piece of DNA that says, hey, make this drug. Or maybe I want to put a piece of DNA into a bacterium that allows it to eat plastic in the ocean. Or maybe I want to insert a piece of DNA that lights up, that makes a protein that lights up when it encounters something like cancer DNA. All of these things are empowered by a paradigm called synthetic biology that we are going to be seeing more and more of in the coming years. If the 20th century was the computer age, the 21st century is going to be the biology age. And what this means is that living things can become factories, they can be computers, they can be sensors, they can be power plants. All of these things can come from synthetic biology, meaning that using biotechnology, we could be getting all the food, fuel, and fiber that we need in order to support the billions of people who live on this earth while still being friendly to the environment and keeping our civilization sustainable for many, many years to come. So where are we going with synthetic biology and other biotechnologies? I found this great quote in The Economist that I wanted to share with you because I want you to take a minute to really picture this. So picture perhaps a world where machines now made of metal and plastic are instead made of flesh and bone. Pets and garden plants can be designed to order. Mammoths, once again, thunder over the tundra. That's a real thing. People are bringing mammoths back. And human reproduction itself ceases to be a matter of luck. This means that using these tools, these biotechnological tools, we have the chance to radically impact life on this planet, including humans. And so again, that's that idea of gene editing. The most famous one of these gene editing technologies that you've probably heard of is called the Crispr system. Crispr is a system that was actually taken, it wasn't invented by humans, it was taken from the bacterial immune system. Humans just figured out how to change it around a bit and use it as a tool. And what we're seeing today is already, this is not science fiction, we are already seeing the impacts of CRISPR on human health. And so it turns out that gene editing comes in two big flavors. The first one of those flavors is called somatic editing. Somatic editing is soma, meaning body. Somatic editing means that you only change the body and that change isn't passed on. So if, for example, picture I had a skin tumor and I used CRISPR to edit the DNA of those tumor cells to be back to normal cells, well, I wouldn't pass that on to my son or my daughter, right? I've only changed my skin. Now, again, this is not science fiction. This is already happening. There are already clinical trials going on today for somatic gene editing. So, for example, there are trials going on for cancer immunotherapies. There are also gene editing trials for types of blood disorders like sickle cell anemia and beta thalassemia. There's a trial going on for an inherited form of blindness where you actually inject the CRISPR components into the retina of the eye. And in one that I think is really interesting, folks are also using gene editing to try to fight urinary tract infections, which can be really, really, really hard to cure any other way. So keep your eyes open for progress in this area because we're going to be seeing very, very soon data coming from these trials. Now, the second flavor of gene editing is one called germline editing. Germline editing is when the edit that you make to the DNA actually gets passed down to the next generation. How do you do that? Well, you do that either by editing a sperm, editing an egg, or editing an embryo before it starts growing into a fetus and then eventually into a person. And as you might imagine, this technique, the germline technique, is far more controversial than the somatic technique. You might remember at the end of 2018, the shocking news broke that 2 baby girls had been born in China who had been edited as embryos. Now, those 2 girls, their names are Lulu and Nana. There's at least one other child out there that nobody is talking about. But this was a huge controversy. There were all kinds of ethical issues that had happened. There was all kinds of scientific questions that still haven't been answered. And what we saw was that the technology was moving much faster than people thought, including Emmanuelle Charpentier and Jennifer Doudna, the women who were did some of the earliest work on the CRISPR system and who won the Nobel Prize just this last year for that. Now, the story isn't over because folks realize that these tools are potentially some of the most powerful tools we have to fight human genetic disease. So how can we use them in ways that are ethical, that are moral, that are embracing our humanity, but are still saying, we've got a really important chance here where we could change the trajectory of humanity for good in a really important way. And however, it's not clear yet how to do that. And in fact, the scientist who was responsible for the experiments that led to those children has been sentenced to prison and will not be allowed to work again in reproductive medicine. So these are super important questions and conversations that we need to be asking right now. These aren't questions for our children. They're questions for us because the time is now, the moment is here. And it's so important for us to have so many voices, men, women, any kind of gender from all over the world having these conversations. Now, I mentioned that CRISPR, the CRISPR-Cas system, a lot of the early work on it was done by two women who won the Nobel Prize last year. So until 2020, it turned out that only 5 out of 184 winners of the Nobel Prize in Chemistry were women. I'm showing you a picture here of the first woman who won the Nobel Prize in Chemistry. This is Dr. Marie Curie. Now, when Marie Curie won the Nobel Prize in 1911 for chemistry, making her the first woman to win that prize, that was actually her second Nobel Prize. She had already won a Nobel Prize in physics in 1904. I want you to remember her. We're going to come back to her in a minute. So last year, when Doudna and Charpentier shared this prize, it was a huge deal. 2 women not sharing a Nobel Prize with a man in chemistry, this area that has traditionally been dominated by men. This was a huge deal. And so go home and tell your daughters that they can win Nobel Prizes too, because this is where we're seeing the impact that women are having on STEM and potentially on, I mean, honestly, I can't think of a more important breakthrough that we could be having. Medicine, agriculture, everything is being touched by gene editing because it touches DNA, and DNA is the source code of all life. So how did Jennifer Doudna, one of the women who worked on CRISPR, how did she get interested in science? So she actually grew up on the big island of Hawaii when she was a kid. And while most of her teachers didn't seem to expect very much of her, she did have some teachers that were interested in her and that were interested in what interested her. She loved hiking, she loved looking at plants and mushrooms, and then she would go and talk to her science teachers about them and say, hey, this is what I saw, I wonder what it means. And she says that one of the most important things that happened to her as a young woman that helped direct her toward a STEM career was her father gave her a copy of a book called The Double Helix, written by James Watson, the man I told you about when we were talking about the DNA structure. Now, I'm showing you here my copy of this book. I have this book. I've read this book several times. And the reason that this was so informative to her and to actually many, many scientists around the world is that the double helix tells the story of science by looking at the people who actually did the science. So seeing scientists as people is something that we're not always very good at. So yes, I own a copy of this book, but actually this is not the book that I would share with my daughter to inspire her to go work on DNA. I instead would point her toward a book called The Code Breaker, which recently came out, which is about Jennifer Doudna specifically, but looks more about the many people, the many men and women who worked on CRISPR and are thinking about where it's going as a technology. You can also check out a documentary on Netflix called Human Nature, where Jennifer Doudna, again, is really prominent in this documentary, looking at how CRISPR and other gene technologies are really poised to impact our life. Now, okay, so I would give my daughter, if I had a daughter, I only have a son, and he's wonderful. If I had a daughter, I would give her a copy of The Code Breaker, but I would also give her a copy of this book. So I am showing you here a picture of one of my prized possessions. This is a biography of Marie Curie, the woman we were just talking about. The Marie Curie biography that was written by her daughter, Eve. Now, funnily, Eve was the daughter of Marie Curie who didn't win a Nobel Prize. Marie Curie's other daughter, Irene, also won a Nobel Prize in 1935 for chemistry. So super impressive family, right? The women all went on to do amazing things. So I would share this book with my daughter because even though it's a bit old-fashioned, there's some stories in here that are, I think, incredibly inspiring and incredibly important. And I've told my son these stories, and now I'm going to tell you one of them because I think they are so important when we're thinking about the impact that women can have in STEM. So this book is interesting because it does tell the story of the early days when Marie and Pierre were doing all of their important work on radium and figuring out all these amazing physics. But it also tells her story after she won her second Nobel Prize. By the way, only 4 people in history have ever won 2 Nobel Prizes. Marie Curie is one, Linus Pauling is another one, Frederick Sanger won for DNA sequencing and for protein sequencing, and then the 4th person is John Bardeen, the physicist. So Marie is in very august company here. So in 1914, the world was heading for war. And Marie watched as all of her male colleagues joined the military, and were dispersed to be working on whatever it was they were working on, leaving her behind in Paris. Now, Marie realized that there was a huge problem. She knew that the new weapons that were going to be used in this new war, in World War I, were going to damage human bodies and human lives on a scale that had never before happened. And she knew that almost all the hospitals at or near the front had no X-ray equipment, no way to see what was going on inside a soldier's body when you brought them off the battlefield. Now, Marie had never actually worked on X-rays herself, but she had taught them in her classes because she was, working at the Sorbonne. So she realized that there was no one else who was going to solve the problem of getting X-ray technology to the front. So she decided that she was going to innovate mobile X-ray units. So she went out and she got some money. She got some funds from people that she knew to fund this new startup venture that she had. And she went around to other French women that she knew, and she talked those women into giving her their cars. So she would go, she'd say, hey, I've got this idea, I need your car. And she always promised that she would give the car back, although she never promised that they would be in very good shape because of what she had planned for them. She got 20 cars donated this way, one at a time. And for each one of those cars, with her own hands, she turned each of them into a mobile X-ray unit. She invented an X-ray machine that was small enough and robust enough that it could be moved into battle. And after she had built each one of those 20 mobile X-ray units herself, she drove them herself directly to the front, directly to the battle. And when she got there, she trained men and women on how to use these mobile X-ray units. So like I said, 20 of them she put together. But that wasn't enough for Marie. She also realized that the battlefield hospitals did not have any X-ray technology. So she figured out how to put X-ray rooms into battlefield hospitals. She built 200 of these battlefield X-ray rooms. And she herself would go into these rooms, take the X-rays, look at the information that was in them, and then work with the surgeons on how they were going to save each of these young men who had come in with a terrible injury. And it has been estimated that between her mobile X-ray units and her hospital X-ray units, Marie impacted the lives of more than 1 million soldiers during World War I. I actually, you can see, I get really emotional when I tell this story because this is the story of a woman who had been fighting her whole life to be recognized by the scientific community. And when she had a chance to step back, instead she stepped up. And she made a huge difference in ways that we are still benefiting from today. Marie Curie is one of my heroes. So I hope I've convinced you that thanks to biotechnology, if the 20th century was the computer age, the 21st century is going to be the biotech age. It's going to be impacting our fight against climate change, protecting our food and our natural resources, and radically transforming our health. And I hope I've also convinced you that women have been making fantastic contributions to these spaces and that way we have even more contributions to give. So how can we help get more women and girls into STEM and technology and empower them to make major lifelong contributions at the highest levels, at all levels throughout? So here are a few suggestions that I have for us. The first thing we need to do is connect. From a very young age, we need to be watching for opportunities to connect our daily lives and the daily lives of our children with STEM and with technology. We need to help girls and boys see that STEM and tech are all around us, making our lives better every single day. And we need to help our girls and our boys understand that every time they use their eyes and their brain and their creativity, they are already being scientists. We need to teach our girls to code. We need to teach our boys to code too. We need to be teaching our people the skills that they need for tomorrow. Our schools are really good at teaching the skills from yesterday, but we need to be looking ahead and saying, what do our children, what do our young people need to learn in order to be successful in a world where STEM and tech are poised to radically impact our lives? So thanks to online innovation in particular, we can be teaching them the skills of tomorrow, even today during pandemic and lockdown and all the uncertainty that we have. So I'm talking about coding, I'm talking about robotics, I'm talking about ethics, design, maker skills. All of these things and more can be learned from a very young age, all the way through your formal education. And even when you're my age, you can be learning to do these things too, all your life. And so that kind of resilience is really the most important skill of all. And I hope you've seen that resilience in the stories that I've told you today about these women in STEM. Because STEM is hard. It's hard for years. And we need to have the resilience and the courage to work hard and to learn hard things so that we can change the world. We also need to keep our eyes open for inspiring stories that we can share with boys, with girls, and with people who are older as well. We're keeping our eyes out for stories that show how powerful and impactful women can be in STEM. Not just scientists and doctors, but entrepreneurs, activists, politicians, and more. We need to be listening to their stories, and we need to be sharing them. And I'd like to invite you to follow me on Twitter and on Instagram. There's my handle. I spend a lot of my time on social media sharing these stories of women who inspire me so that we can go on and do this ourselves in our daily lives. And Argentina, I know you have generations of science women coming up, and it is so important to inspire them to change the world. Now, a friend and colleague of mine, Dr. Jose Mori, has started a media company that's focused on telling the stories of women and people of color in STEM. Now, a lot of these books are meant for young girls to change their perception of what a scientist looks like. And it's to show them that scientists look like them. Scientists aren't men in white coats off somewhere. Scientists are people like you and me, men, women, everyone from all walks of life who are using their minds and their brains to figure out how the world works. So Ad Astra Media, which I'm showing you here, is putting out bilingual editions of their stories. So you can find Spanish titles like Buenas noches, pequeño astronauta, Pequeño doctor, Pequeño astrónoma, and Pequeño veterinario. So I think these stories are great for all ages. I've been sharing them with my son, and I hope you'll have a chance to experience that joy as well, because again, this is a great way to be inspiring the next generation. And finally, we need to be taking a people-centered view, a life-centered view when we build for the future, an ethical view, an inclusive view. Because when we're talking about DNA, when we're talking about biotech, we're talking about life. We're talking about the most precious and unique thing that we have. And as far as we know, there's only one place in the universe where there's life, and that's right here on our planet. So we need to build for an abundant future. We need to build for our generations, for Argentina, and for all people. And so how am I doing this? Well, one way I'm doing this is by talking with folks like you all around the world. But I'm also part of an organization called Homeward Bound, which is elevating women with a background in STEM into positions of leadership so that we are increasing our opportunities to contribute to global issues like plastic pollution, deforestation, loss of biodiversity, and most importantly, the climate crisis, which is going to be an existential challenge that we need to be facing starting today as an entire species. So if you're a woman in STEM, or if you know a woman who is, it doesn't matter how early in your career you are, come check out Homeward Bound. Applications are open for our next leadership cohort. And it's a great opportunity, and I invite you to come join me. And in fact, one of my heroes is part of Homeward Bound. I'm showing you here, this is Cristiana Figueres. She was born in San Jose, Costa Rica. She was formerly the executive secretary of the UN Framework Convention on Climate Change. So she's out there fighting the good fight. And here is something that Christina says that I want to share with you. She says, if you do not control the complex landscape of a challenge, and you rarely do, the most powerful thing you can do is to change how you behave in that landscape, using yourself as a catalyst for overall change. Now, remember what a catalyst is, right? Think like a chemist for a minute. A catalyst is something that makes change happen without being used up itself. And so that's what we want to do. We want to catalyze the future. We want to get as much creativity and innovation in the world as we possibly can. And to me, that means empowering women to program the future of life. So this is how you can stay in touch with me. I hope you will. Remind me that we met at Córdoba Tech Week. Come follow me on social media so you can see these stories and more, and find ways together to get our women empowered to be making these fantastic contributions. Thank you very much. I'll stop there.
Hi, Tiffany. I'm Débora here. Congratulations for your talk. And as a journalist, I could say that we mostly discuss about the lack of women in technology or in STEM careers, but not focus about biotechnology. So I think you are showing us a new challenge to achieve. So I know you are very passionate about translating the leading edge of science into actionable insights for the society. Do you think girls or their parents doesn't know about biotechnology? It's about to create, it's about to solve problems.
Yes, absolutely. I am always surprised when people have this idea that science is boring. Science is an incredibly creative pursuit. It's an incredibly collaborative pursuit. You don't go into science because you think you're going to make a lot of money or you're going to, get a lot of sex and drugs and rock and roll. Like, that's not why you go into science. You go into science because the world around you is so beautiful that you can't stop looking at it, and that you want to find ways to be inspired by the natural world to make daily life better. I think at the very fundamental, most fundamental level, that's what science is. It's a creative pursuit of love, and that's what I want everyone to see.
Yes, for example, here in Argentina, we have a lot of students medicine. I mean, there's more doctors, women, than men. But if we look at another related careers as biology or biotechnology, there's a gap between men and women in this kind of career. Right now, Fabio Grigorchev want to know, isn't it Crispr and their benefits only for rich people?
Oh, so I'm going to respond to the two parts of that. So what you said about there being a gender gap in medicine and in technology, or less of a gender gap in medicine. So in biology, historically, women have been making up 50% or more of undergraduates. So at the undergraduate level, women are very well represented. The problem comes as you move higher and higher up the levels of leadership. At every one of those levels, women drop out of the pipeline. So it's not enough to bring women into STEM. They have to be empowered to stay there. They have to be supported in knowing that they're not going to have to make a choice between family or career, or they're not going to be passed over for a man, even though you're doing the work. All of these things are systemic things, institutional things that we can be working on together in order to make this change happen. And I think what we're going to see is change over time, where first we'll be seeing changes in fields like medicine, like biology, where women tend to be more visible. But as science, as physics, as engineering, as computer science becomes more welcoming to women, I think we'll see those changes start to trickle up and to see a positive feedback. Now, is CRISPR just for the rich? So you guys know, as technology watchers, that anytime you have an early adoption issue, the early adopters are going to be paying more money for a product that's probably not quite as good. So is CRISPR only for the rich? Today, I mean, even rich people can't necessarily get into these clinical trials. So it is, yes. So the short answer is, in the short term, first, I think we're going to see CRISPR being very expensive, and so working for rich people. But you know what? That's not a world that I want to be building. I want to be building a world where everyone has access to these technological breakthroughs, these health breakthroughs, these food breakthroughs. And we do that by deciding together that we're going to democratize these technologies, that we're going to keep pushing to make them cheaper and faster and still be really, really safe. That doesn't happen by itself. It happens because people like you and people like me decide that that's the world we want to build. And I think we can make that decision together.
Agustina Pérez y Carolina Salcedo asked these questions. What experience of yours led you to decide for this technology field?
So that's a great question. So, you know, even when I was very young, I was fascinated by the way the world worked. And actually, my dad was a chemist. And I grew up thinking I was going to be a chemist. And then I remember I spent a summer in my father's laboratory measuring the carbon content of water. Do you know what the carbon content of water is? It's 0 every day. And I thought to myself, oh my goodness, I cannot do this for a living. Fortunately, I learned a lot as a chemist, which was really important for later. But then I realized when I was looking around that I was really interested in living things. And I had actually thought that meant being a doctor, because when I was young, doctors were the only people that I saw doing stuff that had to do at all with biology. And it wasn't until I was in college and I started working in a genetics laboratory that somebody finally said to me, hey, Tiffany, are you interested in the things we already know, or are you interested in the things we don't know? And I said, good question. I'm interested in the things we don't know. And they said, okay, you need to go get a PhD. You need to go do this research. And that was off to the races. Once I started working as a researcher and realizing I had to invent things in order to answer the questions that I thought were interesting, I was hooked for life.
Some experts say that it's going to take at least 200 years from now to close the gap. What do you think about it?
To close the gender gap in STEM?
Yes.
I don't think it has to take 200 years. I have already seen a difference. So again, I'm not that old, but even there's a difference now from when I was in school and what I'm seeing people now. And what I find really exciting, so my son is 10 and his school has some really, really great STEM maker stuff. They have 3D printers, they have robots, the kids are all learning to code. And what I'm seeing in this classroom of 5th graders is almost no difference between boys and girls. I remember when I was a kid being made to feel embarrassed because I knew the right answer and a boy didn't know. I don't see that anymore. And so what I'm hoping is that we can continue to say, hey, it's okay to be smart. Whether you're a girl, whether you're a boy, whether you're something else, it doesn't matter. If you have something to offer, you are welcome here. And I think we can make those changes faster than 200 years because I'm already seeing it happening.
Yes, I agree with you. I'm also optimistic about this topic because it's like an exponential trend, because the children of today should be the STEM professionals of the future. And right now at school, children also have subjects as robotics, coding, these kind of things. So I hope you are right.
And don't forget, we have whole generations now growing up during the pandemic, and they are seeing in real time just how important it is to have science working and how many people from around the world are contributing. This is a potential inspiration once in a generation, maybe once in a century, I hope, moment where humanity gets to step up. And I think of the pandemic as being the dress rehearsal for how we're going to deal with climate change. If we can handle the pandemic, we've got the tools and the mindset and the leaders and the young people who are going to help us tackle climate change.
And right now, there's a lot of companies also talking about this topic because they need talent for doing businesses. And, you know, they need talent, and women, we have all the abilities to do a great career as a biotechnologist or scientifics, or, you know, as well. But do you think when companies talk about this topic, are they talking a marketing slogan, or do you think it's a real issue for us, for them?
Yeah, I think it depends on the company. It depends on the leaders, and it depends on the company culture. I have seen lots of beautiful examples of companies putting their money where their mouth is and getting women in leadership positions, giving them control, and taking away all of these little things that happen over and over to women to undercut them. And I see so many men who are dedicated to being allies, supporting the women that they work with, and making sure that the systems really do change. Now, that said, we have a long way to go. You probably know that, for example, venture capital funds, only a fraction of venture capital funds go to startups that are founded by women. That has to change. And that means not just saying it's important, it means writing the checks to different people. And so if you are an investor, if you are a venture capitalist, I challenge you to be part of the change. Find the women founders, find the good ideas, and give them the money that they need in order to be competing in the market with their male counterparts who have always had a boost over the women. You can be an agent of change as well.
Yes, of course, Anna. On the other hand, I think that women in tech that right now are working in technology should work also as an influencer action, because they have to show us their job in order to motivate to the daughters, the girls of tomorrow. No, so.
It's an interesting issue that you're bringing up, because I want to say, yes, we need to be doing this. We need to be role models. We need to be influencers. But you know what? I'm already working twice as hard for 70% as much pay. And then you put on this another issue on top to say, okay, Tiffany, you also have to do the work to inspire the next generation. And that becomes a really difficult extra burden that women are taking on. And so I'm thrilled when I see women doing that. And I'm even more thrilled when I see our male allies stepping up and helping us with that. Because it can't just be us doing all the work and doing the extra work. We need everybody to be coming to this together.
Well, I think I don't have more questions for the audience. So we can keep talking about this topic for a lot of time, because it's passionate, and we have to fight for the gender equality. So thank you very much, Tiffany, for your time and for sharing with us all your knowledge. And, well, we say goodbye with loud applause.
Thank you all.
Thank you very much. Have a nice day.